Welcome to the lecture number 2 of Descriptive Statistics. In this today's lecture, I'll be taking you guys through the meaning of data, their types, and what do we mean by variables. For example, you must have studied about or heard of the term ordinal variable, categorical variable, discrete variable, continuous variables. But how do these variables really look like in the field of biomedical sciences? Right? Also in the previous lecture, I gave you idea about few of the different kind of the data sets in the field of biomedical world. For example, we looked at scanning based data for example, like MRI data sets or X-ray images. We looked at the protein data sets. We looked about gene expression data sets and so many other data sets as well. By the end of today's lecture, you will be familiar with all these different kind of the data sets and how the data really looks like. For example, if we talk about image, the question that should come into mind is that how do an image really looks like? What is the image really made up of, right? So these are a few of the kind of the questions that I will be covering in the today's lecture. Just a minute, let me share my screen. So fine, so you can see here, we are going to cover the data and their types. Yeah. So see guys, so when we talk about the type of the variables, these are the few of the kind of the variables basically, like quantitative or numerical continuous variables. For example, the first type of the data that we talk about or we get to see in the real world basically is that of numbers, right? For example, when we talk about gene expression data, it comes the format like 1.2, 5 point something, 10 point something, or if it is there in the format of NGS RNA sequencing data, then we have the count data, right? So these are also made up of what? Numbers. So any number, any kind of the data or the variable which have numerical formats falls under this particular type, that is quantitative or numerical variable. Then we have continuous. So continuous means basically whenever between any two interval, we have infinite set of the value. This comes under continuous variable, right? And the opposite of continuous is basically your discrete variable. So discrete is something which can be counted, right? For example, when we talk about NGS RNA seq data, we get the read counts, right? And we know that read counts can definitely be counted. It can never be in figure like a ratio, right? So that data falls under the category of discrete data. Whereas when we talk about the microarray data, here we obtain the intensity of the light, right? So that falls under the continuous variable. Then we have another variable named as categorical or unordered variable, right? So categorical variable is something like, for example, if you talk about the race of humans, if you talk about the different colors, if you talk about the different type of the animals in the data set that we have. So, or suppose we are given a biological gene expression data and we are given a condition vector, right? And the condition vector contains either healthy or cancerous. So this cannot be ordered. You cannot order them, right? And so, and it is something like which falls under a specific category, right? So this type of the data set comes under categorical data basically, and it is a type of nominal variable. Then we have categorical and ordered variable basically, right? So within the categorical variable also, some variables will be like which can be ordered, right? For example, now suppose that you have the color complexity. You are given the color complexity. Now this color complexity, you can also provide a number scale, right? Or for example, like you can see here, fair always lies between good and poor. So this can be ordered. So such kind of a variable falls under categorical ordered variable. And it's a type of qualitative, right? So there are two types of variables, qualitative and the quantitative. Qualitative is something which is going to tell you about the quality of the data. Quantitative is something which is you are able to measure in terms of the numbers. Right? Then we have another kind of a variable named as date or time. Right? So many times we get the data and we have the dates, the time frame, right? So such kind of columns will be considered on the this. And then at the end, we have text data. For example, when we talk about gene sequences, this comes under what? So this will fall under what? Text. Why? Because it's a sequence of the ATGC and all. So it's a text data basically. Now see here. So this is what? This is your text data. And this is what? A gene sequence data corresponding to some particular gene. This I've taken from the NCBI GEO, which I gave you an idea and I showed you in the previous class basically. Then we have this another kind of the data. This is a methylation data. How do I know that it's a methylation data? Based upon the row index ID, right? So it's a methylation providers, and these are the patient identifiers, and these are the values. So in a methylation data, we get to measure the like it's a ratio. It's a ratio of the intensity of the methylated versus non-methylated side, right? So now here we have the decimal values, right? And it's a numeric. It's a numeric and the decimal types. So it will fall under what? Continuous variable, quantitative and continuous. But again, in this data set, you can see one more thing that it is also having some null values, right? So this is another kind of the data type basically, that you might have some null values. Now this null can be there for number of reasons, right? You're not able to measure the value, or these values were not given to you for some reason. It can be n number of reasons can be there. So null means the value is not given to you in your data set basically. And there's a complete science of finding these values, right? So we impute and all. So that we will cover in the few coming lectures. I will give you an idea a little bit. Then we have this scanning image. See, so like this is a scanning image, and this is what in the form of an image. But now the question is, how will this image really look like? So I will show you with the help of our language basically, like what is this image made up of basically. So I'll be taking some another image, but all the images are same basically. It is just what difference in the some pixel value. What do you mean by pixel? I will show you. And then it's another kind of the data set basically. It's what? Signals. So it's a time series kind of the data. It's a time series based data basically, where we are measuring the PQRS waves of the humans. So it's a ECG data basically, right? So there are a few of the different kind of the data sets that we come across in biomedical sciences. Now, what are the different kind of the data types, right? So for example, when we talk about the numeric type, so it might have integer based values, so which comes under like whole numbers. Then we have double numeric or the decimal based value. So these are the data types in our language basically that I'm showing you here. Then we also have complex imaginary number, which you cannot basically, which are not there in the real world, right? So for example, 2 plus 3i and all. Then we have logical data type as well, right? Where we have binary, either true or false, either 0 or 1, right? Then we have character data type, nominal variable kind of things, right? For example, our language, hello world, these are what basically? These are what? Character data types, right? And in R, we always write the characters with the help of inverted commas basically, even in Python. The another variable is basically, or data type is your factor, right? So these are the categorical data with some labels. For example, here we have total 3 different labels specified: low, medium, and high. Then there are few of the special data types, guys. Null value, it is something which are missing in your data. NA values represent missing values in the data set again. NaN value. NaN is, it is not a number. It's in the indeterminate form basically. It's the format of 0 by 0, right? Then we also might have some infinity or the negative infinity values in our data set. So many times you will get these kind of the data sets in your data basically. And based upon all these things, your descriptive statistical analysis will vary. Because if you have text data, your analytical methods will vary. If you have numerical data, analytics will vary basically, right? And then we have there are few of the other data structures based on the data types. For example, vector, right? So vector is what? It's a collection. When we have more than two elements, we need to store them into a single some list kind of thing. So we come across the concept of vector in R. Then a list. Now, if you have to store heterogeneous data, suppose you have total three different variables, and you need to store all those three variables together into a single container. So there you come across list concept, where these three variables can be different data type. Then we have matrix. So matrix is what? It's a M cross N or 2D array, basically. It can be 1D array as well. Right? So matrix is what? It's a 2D collection of the same data types. And one more thing. In matrix, you will have all the values of the same data type. Even if one single value you have incorporated as not decimal, but character type, character, then all your numeric values will get converted to the character type, basically. Then we have data frame. So data frame is what? It's a tabular data, but you can have heterogeneous variables as well in a data frame, which is not allowed in a matrix. And the array is what? Multidimensional data structure. Now, this is a question that we should ask by the end of this slide now, that what is the difference between a matrix and a data type, right? So very simple. Matrix, all the data values need to be the homogeneous in nature. All values have to be of the same nature. Whereas a data frame can have heterogeneous data types, basically. Now, let me show you a few of those things with the help of R language, guys. Okay. So I will show you both in R and Python, basically. So let me present my screen. Great. So, fine. So, guys, you can see here that I'm going to like, for example, let me show you this image first. What is the name of the image? Okay, great. You can see here, guys, that, sorry. Yeah, see here. For example, we can see this is an image of a cat, right? It's a cat. But now, question is, how do this cat really look like in terms of analytics? Because we have to do some analytics, right? So how do you do analytics upon this image, basically, right? So for that, let me show you how we basically read these kind of the images, and these exist in the field of data science, basically. So for that, what you can do basically, guys, is that you need to install a package named the image R. And after that, after installation is done, you need to call the library image R. Then here I am writing a code load.image where I'm going to load the image, and I'm mentioning here the path of the image, basically, right? And now my image is here. So now you can see here that fine. So when I typed an image, it is telling me it's a image of width 500 pixel, cross height 333 pixels. For example, let me show you. So dimension, it's a matrix basically, by the way. See. So now you can see here that it's an n dimension array, right? So it's a 4D array basically. Okay. Where these two represents your pixel values, and this is the channels. This is the channel. There are three different channels basically, R, G, and the B, right? Now suppose that you need to look at this image. So what you can do, you can type a command called plot img. Okay, now you can see here that your cat looks like this. So this is how you can load the image of a cat. Okay, and now suppose that you want to apply some conditioning upon this image, right? And I'm applying the conditioning that I need to draw a box, or I need to shade those regions where the image pixel value is lesser than 0.5. Now you can say, fine. So I've applied a filter here. And now I'm going to draw my image and then create a box around this value. Just a minute. Sorry, yeah. See. So now here I have passed color green. So now you can see here all the places where this green color marking is there, the pixel values will be less than 0.5. Right? So this is just a little bit idea, like how do image looks like. Similarly, we have the scanning based images. Those also are present in this format only. That is the images, right? Now, let me show you one more data type basically. Now see here. Now it's a gene expression data, guys, right? So here you can see that the first column, we have the samples, the patient IDs, and then we have the gene names. And the last column is the condition. Now here we have different data types. Like we have these numeric values and we have this categorical variable. So is it a data frame or a matrix? Try to answer, right? Because heterogeneous data is there, so it's a data frame, right? And now suppose I want to get an idea about this condition. Like what are the different kind of the conditions there, right? So again, I will apply a function called counter in Python basically and get the idea that what are the different kind of the categorical types present here. And there are two classes, lung adenocarcinoma and lung squamous cell carcinoma, right? And both having the frequency of 576 and 533, right? So this is all about for the today's lecture, guys, right? So in the coming class, now we'll be starting with the proper analytics, right? So now you have a little bit idea about how do our data looks like, right? And now in the coming lecture, we'll be starting with central tendencies. So I will be giving you the idea about what is the meaning of mean, mode, median, how you can calculate this with R or Python, right? And then we'll be starting with proceeding with few the advanced concepts. Thank you so much.